This is my first slide. I am Dr. Ajit Kumar Saxena, and genetics exposure challenges in prevention and healthcare delivery in developing country like India. And I am sharing my work that I worked for the last 10 years in Bihar, poor state in India, as professor. The next, uh, the congenital anomalies are highly complex uh, disorders leading to trigger abnormal organogenesis. Challenges are both for scientists as well as clinicians to cure it. Genetic and epigenetic factors are responsible for such anomalies that lead to the birth defects. The etiology is highly complex because of complexity of the human genome involved at chromosome level or single or multiple genes or both. If only chromosome involvement that lead to the mental retardation syndrome patients with high frequency in Bihar states. The frequency of another neurological disorder is neurotube development. Down syndrome patient that varies different in different populations and in different uh, socio-economics problems. And this is my third slide.
Change. Correct.
This is my third slide. No. Previous one. Yes. This is a collective data of the type of abnormal patients, understanding the genetic diversity on my registry, uh, and setup and maintenance of the policies. If you will look in between, there is a family having a small child of 3 month old mental retardations. On the extreme right hand, you will see there is a baby with having the ambiguous genitalia. And in below, you will like to see the Down syndrome. This is not Down syndrome, but it is a syndromic of mental retardation patients. And the variety of another other than abnormal is the gynecomastia. This one is a gynecomastia, and she is suffering from the amenorrhea patients. And similarly, this is a Klinefelter syndrome. So this is a group of my collective patients data. Now next one. Next slide, please. Yes. The type of most common congenital anomalies is the Down syndrome patients. No, previous one slide. Previous slide, please. Yes. The type of common congenital anomalies are associated with genetic disorder in Bihar based on genetic registry which I have been developed from 2014 to 2024. Neurological disorders which having a clinical phenotypic changes depend upon the degree of genetic damage in individual and their severity for their lifestyle. Next slide, please. Yes, this is a clinically Down syndrome patients, and the frequency of these Down syndrome patients amongst all is 12.04 and having a trisomy 21. I am showing in the next slides. Next slide, please. Yes, the first step how to diagnose the genetic patient is to develop the G-banded karyotypes. And karyotypes, if you will develop, you will find a extra copy of chromosome number 21 on the G group chromosomes. You can see that there is an extra copy of the chromosome 21. So the patient or the case confirm that he is a Down syndrome patient, and when we have done a painting chromosome painting, we have a software and confirm there is a extra copy of the 21. So this is a case of the Down syndrome patients. Next one, yes, this is a we have used fluorescent in situ hybridization using a specific probe of trisomy 21 to further confirms of the above findings of trisomy 21 chromosomes, and in interface we have seen 3 dots instead of 2. So the extra copy of the chromosomes are appearing using a probe and by fish analysis. So these are the facilities in my laboratory for the early diagnosis of neurological disorders for the patients. Next, yes, this is not a trisomy patient, but it is a syndromic mental retardation patient. The frequency of these patients are also very high. And when we have developed the karyotypes in mental retarded syndromic cases, we have found a different and variety of karyotypes. For an example, 47XY or 47XX. Mosaicism were also observed in case of female cases, 46XX or 45X0. The another case we have also observed in case of female, the fragile sites. Fragile sites are the highly rare type of individual genetic variations, and XQ27.1 is a breakpoint which is found on the sex chromosome, X chromosomes. And the another novel finding which I have observed in this case is, no doubt there is a 47X case, the Y chromosome shape has been changed into the ring chromosomes. So this is one of the most important case, and I have seen that the ring chromosome of Y that break and reunite to form the Y chromosome associated with the syndromic cases of the mental retardations. Now we come, next slide please. Yes, this is the another category of the neurological disorders. The this after fertilization, gastrulations, the organogenesis start, and if the antenatally exposed with the tetogens, that first affect to the first organogenesis, that the development of the central nervous systems. If you will see that the anencephaly or exencephaly, these first two categories die in situ. This is the severe category of the neurological disorders. And the another category is the myelomeningocele, the cephalic regions just behind the neck, then cephalic thoracic regions, and then cephalic regions just behind the back of it and in this. So this category is involved both genetics, involvement of variation in the structural variation of the karyotypes, number one. Number two, there is a single gene mutations in the folate metabolisms. The folic acid deficiency in the parents might have increased the risk for the development of neural tube defects, and for the clinicians, it is very difficult to save the first category. You will see the anencephaly or exencephaly, they die in situ, while the myelomeningocele cases, they also very difficult just behind the cephalic regions, difficult to operate by the pediatric surgeons and neurological surgeons. And the incidence of mortality is very high. But the thoracic and cephalic region, we try to save it. The next slide. Yes, the neurotube defects are the complex disorders that show the karyotypic variations and the mutation in the folic acid metabolism MTHFR C677T gene polymorphism increased risk factor in eastern population of India. This manuscript we have published in 2012 in Genetics and Molecular Research. It is a PubMed paper, and the beauty of this work is out of 10 publications, uh, my paper was top in 1 out of 10, and CCMB and Pune was exist in uh, 6th and 7th. So this is a beauty of my work, and this work was supported by Government of India Department of Technology, Biotechnology. Now we are coming to the another neurological, other than neurological disorders. Please change the slide. Yes, this is the hypospadiasis, and the frequency. This is a birth, uh, abnormal birth, where the perforated penis is there, a arrowhead, and the frequency of hypospadias is 4.69. This is the second category of abnormal sexual development, where the perforated penis is there in large number of the patient who referred for my laboratory for early diagnosis and find out the cause of it. Being a scientist, so we have seen that the karyotypic variations were uh, trisomy 18 were recorded along with 21, and mosaicisms were also reported, and monosomy of loss of Y chromosomes we have reported, and 20. So there is a gain as well as loss of chromosomes were reported while preparing the karyotypes. And the most important finding is the loss of Y chromosomes. And we cannot reach into the conclusion that how the Y chromosomes play important role for the perforation of the penis at the bottom. So, and the another important findings were reported. There is a chromatid break involving chromosome number 3q25 region. And when we recorded and observed by using the automatic software, and found there is a loss of 25 mega base pair DNA during the breakage. Third finding we have reported, WT1 gene mutations is also involved in it, and hypospadiasis. The finding we have not yet published, but there is some involvement of fibroblast growth factors, androgen receptor, or endocrine dysfunction might have responsible for the abnormal differentiation and development of the hypospadiasis or perforated of the penis. The incidence of uh, the hypospadias cases are very high in Bihar. So I might have thought that in Bihar is a poor region and everybody expected to be a male child. So when they came to know, they have knowingly, unknowingly have taken a medicine that causes the abnormal development. Next slide, please. Next slide, please. Yes, this is the another category of the uh, abnormal development, that is the cryptorchidisms. Cryptorchidisms, the frequency of total cases was 1.28, still high, and you will see the scrotum region where the arrowhead is there. There is a complete loss of testicles, either both or unilaterally. And when we develop the karyotypes, they showing XYY, the extra copy of the Y chromosomes, number one. Number two, there is an extra copy of 21, that is trisomy 21. And the monosomy, the loss of 16 chromosomes. So these are the karyotypic variations in the cryptorchidism patient where the testicles is not present in the scrotum. Next, please. Yes. The another relevant finding that the 60% cells of the cryptorchidism patient, the centromeric damage was present in the uh, metaphase cell. If you will see this metaphase after silver staining, you will find the centromeric regions are having a clear breakpoints. So I reach into the conclusion that the centromeric regions are highly conserved in nature. So that might have played a significant role during the organogenesis or development and differentiation of the testis. Result, the testis failed to reach into the scrotum. If you will see that, there was a no swelling. Next slide, please. Next slide. Yes. So we go up to the depth of it. We have used a WT1 gene RT-PCR based analysis found the mutation of WT1 gene in the cryptorchidisms as well as the DNA copy number variations. If you'll see, there is a large number of cases are there, and you will find the variation of the frequency of DNA copy number variation of WT1 genes. So these are the genetics factors that are responsible for uh, increasing the genetic susceptibility. The next slide, please. Next slide, please. Yes, this is a very important slides. In the case of these cryptorchidisms patients, the stem cells, role of stem cell, because you must be knowing during the organogenesis, the stem cells play a significant role for the development of normal organs. The stem cell, we have used a marker of SOX9A. You will see by the RT-PCR that the number is decreasing or completely diminishing of the SOX9A in this patient number 7 or in 9. This one when we compare with the control, and similarly the copy number variations. So these are the two very important findings, and I have published these two findings in Genetics and Molecular Research. The online impact factor is 92.3 in the PubMed. Next slide, please. Yes. Now we come to the another category of the abnormal development, that is the ambiguous genitalia. If you will look this patient, which having a arrow, this is have both the genital organs. You will see that both penis as well as the vagina perforated is there. So this ambiguous genitalia is very difficult to early diagnosis in these three patients. But we have found a novel karyotype that there is a translocation between G group chromosome, D group chromosomes. You see that. And the frequency of this patient is rare, 0.74. Now we are sharing another very important, please next slide. Yes. I am going to share the another abnormal development, and the incidence was very rare when I was in I left All India Institute of Medical Science, New Delhi, and joined in Bihar, All India Institute of Medical Sciences. I found the 0.7% is anorectal malformation. When the baby is born, there is a closed rectum. So first 2 3 days, the mothers are unable to identify it. But after that, when they come in contact after 1 week, then they show there is a rectum is closed. So the anorectal malformations is another type of genetically abnormal differentiation and highly complex congenital malformation of the hindgut and stenosis and perforated anus. When we develop the karyotype from this neonate, we have found the unbalanced translocation involving chromosome 6 and 9, Q23 and Q34 regions. Beside these structural variations, we have also seen monosomy of chromosome 18 and deletion of chromosome 18 PR and inversion of 14 chromosome number. And we have also simultaneously, because these are the uh, neonates, so we have also simultaneously studied the stem cells, because the stem cell is the early transcription factor and they play a very important role during the organogenesis. So we have seen the mutation of OCT4, because there is a three important stem cell: SOX, OCT, and NANOG. These are play a very important role during the organogenesis and maintain pluripotency. If any of the unknown tretinogen causes mother is exposed unknown tretinogen antenatally, this fetus causes mutation in the stem cell that might have increased the abnormal development of the organs, like anorectal malformations, OCT4, 577 base pair gene mutations. And we have also found the point mutation after Sanger sequencing of the nano, that showing the G change into T and followed by guanine change into the thymidine. It is a point mutation. And we have also studied the folic acid metabolism, the MTHFR C677T polymorphism showing genetic heterogeneity due to the change of cysteine into the thymidine, C change into T. So this finding we have also published of this anolectal malformation cases in 2022. So these are the variety of the abnormal sexual development and that leading to the birth defect. And next slide, please. Yes, the here is the one region near the bank of the river. In these cases, we have found the congenital deafness. All family members in the family are suffering from congenital deafness due to the mutation of connexin 34 genes, as well as there is some defect on the stem cell. And the third, the another anomaly is the craniofacial anomalies, including cleft lip or with or without palate. So these are the two which is also been recorded in my genetic registry, and I am unable to find out the findings, but I have reported in my book. I am going to show you later. Next slide, please. Next slide. Yes, this is my contribution for the last uh, 2 decades. I have published the books Gene and Male Infertility, including Novel Coronavirus 2000 and Fish Analysis of Drugs and the Chemicals. These two books are from Springer, and this is from Science Index. A recent book is I have published this one, the gene, the basic concept of genetics, basic and basic and clinical consequences of genetic disorders in the human. Where from genetics, from this is book where we have showing every clinical aspect during the development from basic to the. This is from Cambridge. So the next slide, please. Yes, I am going to set up a cancer genetics laboratory where I recently joined, and that's the recently joined as a director for the cancer patients, because it is very difficult to diagnose the early diagnosis of the cancer patient based on acute myeloid leukemia to uh, chronic myeloid leukemia by using uh, four probes. So this is my under development process in the field of molecular genetics in whole of the Bihar region, including Chhattisgarh and Jharkhand. The next, you must, next slide, please. Yes. After my study of the several years and genetic registry, one question always struck in the mind: what are the factors why the high incidence of abnormal congenital malformations leading to the birth is high in Bihar? So I found that the arsenic in water, the arsenic contents are very high. So that 12.4% was the highest frequency of the mental retarded, the Down syndrome patients were there. And you must be knowing that the baby is not, but the whole family become chronic to see such type of the patients, as well as the society become also very difficult to tolerate it. So we have seen that the in vitro experiment we have done that water content is the arsenic and the whole food chain is affected. Does arsenic causes the mental retardation? To answer of it, my students have used arsenic of the Down syndrome patients, and PCR based have analyzed the DNA copy number variations of SOX4 genes, because you must be knowing SOX4 is a early transcription factor during differentiation and development of the neurological or central nervous system of the neurons, and confirm increasing risk factor if the cells are exposed in vitro with the arsenic. You can see the intensity of the bands in increasing systematically. Similarly, the copy number variations is too. So that might have increased the risk factor for the development of Down syndrome patients. So these are my study for the last 1 decade in my stay in Bihar. Is a poor states, and I am also developing public-private mode sector to genetics laboratory for the first clinical diagnosis, postnatally, then antenatally, then we will provide the systematic management for it, and we are also going to set up a basic clinical research associated with this congenital malformations or birth defects to reduce the incidence of the frequency and get better for the Bihar populations. The next slide, please. Next slide. Yes, I would like to thank to all the viewers and listeners patiently. And my setup, my laboratory was inaugurated by Dr. Lalji Singh, father of DNA fingerprinting technology in my country, and we celebrate he is no more. He was the former director of Center Cellular and Molecular Biology in Hyderabad and vice chancellor of the Banaras Hindu University. And there is no limit of teaching and learning. What he suggests to me, I see 3-month-old baby is sitting in the DPS teachers that suggesting I am not going to read A to Z 26 alphabet. I will read only 5 alphabet: A, C, adenine, cytosine, G, guanine, and thymidine, and last uracil to stop. So thank you very much for the audience, and thank you very much for the organizers, and especially thankful for organizing those Malvika ji and other Jennifers who was continuous contact with me, and I am happy and congratulated.